So this morning, I'm very eager to introduce our speaker, Dr. Jason Bond. Jason will be presenting soybean foliar and stem diseases. Foliar and stem diseases and insect pests are among the greatest threats to soybean production, and I'm a plant pathologist, so I definitely believe that. Research efforts and attention have been primarily focused on the health of foliage, pods, and seeds, even though many pathogens and insects compromise the health of soybean systems. And this leads to yield loss. And so we have funded research with Dr. Bond to try to understand what pests and pathogens are out there and make sure that our yield is staying in the field. Dr. Jason Bond is a professor at Southern Illinois University-Carbondale and the director of the Illinois Soybean Center at SIUC. And his research and teaching program focuses on disease management in row crops in the Midwest. And he is also the advisor for the Agronomy Society at Southern and a second generation plant pathologist, which is pretty cool, and was raised on a watermelon farm. So you want to sneak some of that in at the end, that might be kind of interesting. So Jason is available for questions in the presentation. For our folks online, we do have a moderator. So if you have questions, drop them in the chat and we'll get them answered here. Jason, if I could just have you repeat any questions that get asked in the room, that would help us out a lot. And here is the clicker and the floor is yours.
I think I need to stay somewhat close to this microphone, but I might wander away. 11:14. I think we end at 11:55, but I looked at the agenda, and if you notice that lunch starts at 11:55 too. So we have a little bit of and I think our room is the furthest from the food. So we're going to talk about expectations and responsibilities. What my wife is always preaching to me, usually it's focused on me, because people need to know what the expectations are and the responsibilities. So the responsibilities are going to rely on Megan to stop me from messing up your transition to be somewhere in the middle to the front part of that food room. Okay, so she's going to stand up and tell me when we have probably about 10 minutes left. So 11:40, 11:45, something like that, to help me to stop. When we do questions, we have rewards. I don't see any of my grad students in here. There's Leo. So Leo is one of our grad students. He's going to be presenting his research today on one of the posters. Some very important work on soybean systematode. A great way to minimize its impact on soybeans with something a lot of us already do. We plant wheat before soybeans. So go and watch his presentation. It's very interesting to see how that impacts SC. And now I'm getting into his talk and not focusing on my talk. But Leo is going to, when we answer questions, we have books up here to reward you for your participation. We have the Farmer's Guide to Soybean Diseases. This is an excellent resource put out by the land-grant universities. It's probably about a $20 book if you go online and buy it. So that should be some encouragement. We also have a Farmer's Guide to Corn Diseases in the stacks over there. So Leo, when we start answering questions, is going to go and get the book and hand it to those who ask questions. For those of you, but more than probably just Megan and I, but really like plant pathogens, we have the Compendium of Soybean Disease and Pests. This is now I didn't pay $129. I don't want you to think that we're this wealthy at SIU that I go buy books and go hand them out at meetings. I got these on a discount a while back, but if you went online today, you would pay a minimum of $129 for this book. Now, this book's going to tell you a lot of information about the pathogens that are also in the management guide, but they're a little bit more focused on things like, well, it covers everything else that would be in that book, but things like the spore size and how it produces these fungi reproduce. And some of that's kind of, pardon the pun, in the weeds a little bit. Maybe for some people may not be interested in all of that information, but it's an excellent resource. Very comprehensive. So for those that ask questions, you might even get one of these. So we have 3 of these. So the pressure's on. So after we get through 3 questions, I'm not sure if we still will have one of these available. So unless we can get it out. Okay. So my presentation today is going to be focusing on both soybean foliar and also some of the stem diseases that we deal with here in the state of Illinois. Most of them are continual threats to production, primarily because they overwinter in our fields. even maybe not a particular field that you're growing your crop in this year, but maybe a neighboring field or your neighbors. And sometimes our southern neighbors like Kentucky and Tennessee, what's happening in their fields really contribute to what we deal with as these pathogens will move into our areas. Okay. Just to give you, it's been a while since I've been here at the Soybean Summit. It's a great honor to be back here and see so many faces that we've seen before with a lot of weather challenges that we've had over the years. This is a fantastic day to be able to come and gather, and hopefully we're on the back end of this epidemic that's prevented us from being able to interact to a great degree. At SIU, just kind of reintroduce who we are, since there's many of you that have not met before. Just to tell you a little bit about the plant pathology program at SIU. It's actually both a combination of applied and a basic research program. So my colleague, Dr. Ahmad Fakori, who is not able to be with us today, he and I work closely with all of our grad students, and most of our projects have a very applied focus in some capacity to how do we manage these organisms. But in many cases, another component of many of these projects is a greater understanding of the biology of these organisms. What's so unique about them that's going to make them be a problem for us to manage in future years. Some of the research projects that we have ongoing right now, quite a lot of work is done focused on these soil-borne issues. Our biggest issue in terms of plant pathology in Illinois, and really anywhere you grow soybeans, is soybean system effect. That is a very challenging pathogen, and you could spend an entire week just trying to talk about all the complexities of that particular organism. So we have various projects that we're working with ISA and also with the USB and North Central on trying to come up with better management options for managing this important pathogen. We also have quite a number of projects to look at all the foliar pathogens that we deal with. They rob a large amount of yield, not only in Illinois, but across this country. But one area that's sometimes kind of forgotten is that the health of the stem. And that's an area where we have not only some insect pests that cause great losses and issues when we get ready to harvest, but we also have a lot of pathogens that, in addition to attacking the foliage and maybe our pods and our seeds, but will also cause great damage to our stems. And so some of the most common things that we would see in the state of Illinois as we think about some of the foliar diseases, one of the ones that I come in contact with almost every field, if we're having reasonable growing conditions, it's very easy to find this one disease called downy mildew. You see the chlorotic spots on the leaves, you flip the leaf over, that's how it gets that name, downy mildew, because on the underside or the downside of the leaf, you have this sporulation. So it can look pretty dramatic in the field, but generally, I've never seen a case where if you sprayed a fungicide, where you control that pathogen, that you get a response in yield. So it does not, for whatever reason, with the genetics that we grow and in our environment, it is not a yield robber. It is a parasite because it attacks our soybeans and it causes symptoms. So it is you can call it a disease-causing agent, but it rarely would impact yield in our areas. Generally, in other parts of the world, it does cause some yield loss. Septoria brown spot is another one that is in most fields. It's usually easy to find it as that plant starts shutting down as it approaches maturity. And when that happens, a lot of things start popping out on the leaves and are pretty evident. It is a yield robber in some environments. And so in some cases where you see people spray a fungicide, they do see a response in controlling this pathogen. This other disease called Cercospora blight, it also can attack this pathogen as it attacks the leaves. If it attacks the pods, infects the seeds, it will lead to a disease that we call purple seed stain. So probably you have seen that to some degree. It's pretty easy to see. It's pretty widespread. The fact that it causes this seed discoloration also tells us that it is seed-borne. So we can move it around the country. We can move it from continent to continent as we move soybean seed. So it is a pathogen that we can actually bring to our fields if it's not there yet. Typically, it does cause yield loss. There are differences in varieties. We see this quite apparent, especially with a big variety trial. You will see how the varieties respond to this disease differently. And so it does also impact yield. One of the complexities of this pathogen is that the timing of when it arrives and it infects those leaves, you don't immediately get symptoms. And so you don't really know it's there. Then it starts kind of showing up as the plant goes through those later reproductive stages. And so timing of a fungicide application is where that one is pretty difficult. And it's not real clear how much yield protection you get when you spray these fungicides, mainly because timing is very, that's just not, it's just not as easy as it maybe is with some of our other foliar diseases. So you don't ever normally see recommendations, at least in our part of the country, for spraying fungicides for Cercospora blight because of this. But in North Carolina and other places in the southeastern United States, they're under a massive disease pressure with this disease. It's probably one of the more challenging ones in the state that I grew up in Louisiana. This next disease, which is caused by a closely related pathogen, it's called Frogeye leaf spot. The pathogen is Cercospora surgina. This pathogen is widespread in our region. It overwinters in residue in many parts of this region. So it's pretty ever present. What dictates if you have it or not is generally our environment. For whatever reason, 2022 was a pretty light year for Frogeye leaf spot in Southern Illinois and also into Kentucky and Tennessee. If you remember part of that season, we had a real dry period in Western Kentucky and also in Tennessee. And so that kind of slowed kind of the disease. So it wasn't, it was widespread in Illinois, but it was localized really on its impact in terms of how much disease was there. So just because it's in the field does not mean that necessarily yield is going to be lost. It needs a certain level of severity. And so it gets the name Frogeye leaf spot. I have to try to explain this. I don't really see it when I think about the plant, but it's the terminology is because of the little brown lesion that you see is surrounded with this little black border, and it's supposed to kind of mirror that of a frogeye. So maybe y'all see that, and there's a lot of different species of frogs that you might have seen, but I don't normally see that as easily. When you look at this picture on the left showing in the canopy, when the disease pressure starts getting very severe, this was taken in Southern Illinois, this picture, the leaves, as they start getting under intense pressure, they start, you can see the ones in the bottom or the mid canopy starting to yellow a little bit. And then once that happens, they usually start defoliating. So yield loss can come from not only the pathogen infecting the tissue and causing these lesions, and significant yield loss can occur just with that. But once we start getting in the defoliation stage, that yield loss starts ramping up even more. You can even have the pathogen under the right environments. If we have enough moisture and humidity, it will attack the pods and can even infect the seeds. And so you're even compounding those losses. So it is a major yield robber. I'm not seeing some of the little things that my thing here. Let's see. Leave it to the professor to come in and start having all kinds of. Yeah, I think I need to know that. Okay. So I really didn't need that piece of information. I already said it after all of that. So the third point there are strobilurin resistant isolates of this pathogen, and it's pretty widespread. I think it was maybe 2004, 2005. When Carl Bradley was here at the University of Illinois, they were starting to get the first reports of this pathogen, not only in Southern Illinois, but also in Western Tennessee, being resistant to the strobilurin class of chemistry. And so basically today, wherever people are sampling to do to see if their isolate is resistant to the strobilurins, it's a yes. So it's all across the country. So to determine does my field have this or not is probably inconsequential because most of the isolates have been tested in the state of Illinois. A great percentage of them are more resistant to this chemistry. So never do we manage it anymore with just a single mode of action. So strobilurins are great chemistry and are still very important for use in both corn and soybean, but not just by themselves. So we need two modes of action, and we see some companies now even going to 3 modes of action. This is one of my first questions. I know I see some people in here that's got an entomology background. We also have a mixture of people that have managed both the insects and plant pathogens and also plant pathologists. And so my first question is on the little simulated insect feeding. These are some little neat teaching aids. I think I saw some out in the lobby that were available. And so I'm looking at the one that says 30%. And this always boggles my mind when I look at the growth stage of R1, R2, the threshold would be 30%. I mean, as a plant pathologist, if I had this kind of feeding, I would be losing my mind. But I know that, as it turns, we have to have a certain amount before it justifies spraying. But if you look over to the leaf spot of Frogeye, the image, that's pretty good level of infestation on that leaf. But maybe it doesn't look as bad as that number 30 of the simulated insect. And so why would this insect, when it's causing that feeding, why does it need to be at 30%, but yet we can have such less symptomology on the soybean leaf and still cause yield loss? If we're just comparing the size of the spots and the amount of the spots, why do you think on the disease sample you would have yield loss even before you hit the 30% or even at that threshold? Hope I asked that question right.
Stress.
Stress, that's a good thing. What else? What could lead to greater stress on the leaf that's being attacked by that plant pathogen versus the insect feeding?
The systemic effect versus just loss of surface area.
That's good. So you have a kind of systemic, but what else is going, what would cause that systemic effect? Leo, you probably need to move to the front and get some books ready. So, what else? So in both cases, we're losing some photosynthesis. The shot hole where the insect is fed is gone, and also that dead tissue, we've lost photosynthesis. But in this, for disease, those spots aren't technically dead. Even though they're dead tissue, there's still life in that lesion. What do you think that life would be? Oops. So on this, I think my pointer's gonna work. Let's see if I can do this. On this lesion, all this brown tissue, you see this little white sporulation? So that's the organism reproducing. And so for that organism to produce these spores, that energy has to come from somewhere, and it's coming from the plant. So loss of photosynthesis and a continual draw of energy from that plant. And it's not a one-time event when it produces these spores. It's going to produce these spores, they're going to be liberated, go to other parts of the plant, other parts of the field, other fields, neighbors, and so forth. So this is a continual thing this pathogen is going to do, possibly for 20 to 30 days of continually reproducing or producing more copies of itself with these spores. So that's a draw on the pathogen. So you get a book. The stress guy gets a book. Which one did you want? Leo? I'll let you handle that negotiation, okay? Who else had that? Who else had the answer? Someone in the back too, so catch him on the way out. So. No, you can give them right now. All right, so to manage this pathogen, we have options. So it's not, do we just have to react, which is not necessarily when we just spray a fungicide, but if we're going and scouting the field and we see it, we actually are reacting to the pathogen being present or the disease being showing symptoms. So there are options we can take well in advance of that. So when we rotate our crops, to anything that's other than, if we have soybean, if we rotate that field to a non-host like corn, wheat, other crops, that is going to reduce the amount of inoculum in that field. The pathogen is going to live in that residue to some degree. And so that helps reduce, doesn't eliminate the potential for the pathogen coming, but will help reduce it. The other thing that we can do is we can choose resistant varieties. Now, most companies, when I look at their varieties, they are always marking some of those that have resistance to Frogeye leaf spot. And if you don't mention it, then it's probably not been tested, or maybe they're not really that curious or interested to tell you that it's susceptible. But you can read these brochures and get some pretty good guidance or work with a seed advisor and deploy resistant varieties. That will go a long way to helping manage this disease. The other thing that we commonly, and so I know there's a lot of resistance because as a plant pathologist to find that real sweet variety to put in your disease trials is getting harder and harder because the companies have done such a good job, and the universities, at deploying this resistance. There are tolerant varieties out there. And so I have this picture that Laura Campbell gave to me. She works with Corteva. And you can see those spots look like little mini Frogeye lesions. Now, if those lesions stay that same size for a week or more and don't expand and continually get larger and coalesce into other lesions, larger lesions, then that's a type of resistance response to this pathogen. And so you can have a plant that has tons and tons of these little tiny lesions, and you can spray a fungicide and have that check, and you can make that plant look a whole lot cleaner. And in many cases, you will not see a yield response because those varieties, while they may show those symptoms, they are not, they're a tolerant variety. They do not have that same level of susceptibility as a susceptible variety. So you do come across that. I have experience with that, where we look at those variety brochures and find some of their varieties that they say are the worst for Frogeye, and we've planted them, and we get disappointed as a scientist to try to study them, just to find out that variety does have some type of resistance that's not described, or the company doesn't know is in that line, or it could potentially be tolerant. So there's some variability that we see with this pathogen. So my question is, when should you spray for this disease? Based off of information that we've seen, when we spray foliar fungicides, what do we most commonly hear? R3. What else? Any other timings? Sometimes you'll hear conflicting information on that. They're not really conflicting, but you'll hear different information. Sometimes you'll hear R4 or R5. And so sometimes if you're producing soybean seed and their seed quality is very important, and you're dealing with pathogens, or you're trying to control pathogens that will not only infect the foliage, but also attack the seed, then you might have guidance from that, whoever you're increasing that seed for, to spray at a later date. But generally in the literature, so this guy gets a book, you already got a book. Oh, yeah. Oh, you're good shape. So the in the literature and most research shows that R3 timing is really the best. But I'll tell you, most times when we go out into fields in Illinois, even in our disease hotspots, R3, you don't see much foliage, much Frogeye leaf spot. It kind of comes in a little bit later. Now I say that word comes in a little bit later, and I would fail a plant pathology question or an exam if that term coming in later, because the pathogen is already there at R3, or sometimes even earlier. But what we're looking for when we go out and search these fields is actually symptoms, these little spots, not the pathogen. And so for this pathogen to produce this spot at R3, it would have had to infect 12 to 14 days prior to that. When spore lands on the leaf, gets into the plant doesn't let us know that it's sick for at least 12 days after that process takes place. That's called a latent period. So a lot of things can be happening in your field during the period of time when we're not seeing the disease. So it's helpful to, when your plants are sick, for us to know as soon as possible. Same thing for like with COVID. We had people walking around spreading, infecting because people weren't showing symptoms. Whereas if you come in contact with the flu, his sneeze was on cue. But if you come in contact with somebody with the flu, usually like in a 12 hours, 24 hours, you know you got sick, and you probably know who gave it to you, you know. Differences in latent periods. So these pathogens all have these different timings in which once they infect the leaf before you start seeing those symptoms. That makes it challenging. And so under the right environmental conditions, you can see one spot and you're like, a few leaves. I wonder if I should spray. Now I've gone to the part of the presentation where I need to move this down. So this is what we've seen in some real hot spots. Now, this is not indicative of all fields in Illinois. I had an extension person at the state of Illinois back maybe 20 years ago that told me you cannot go and doom and gloom and tell people there's no hope, you know. And so, and also this is not expected in every single field. Okay. But now in these hot spots, where we've seen Frogeye from year to year, maybe some production practices that really drive it. This can occur. And as we go a little bit further south from Southern Illinois, going to Kentucky and West Tennessee, this is a very real thing that happens very frequently, almost every year. So within 10 days of what that application date, so you make the decision not to spray, that's the kind of explosion you can see. So most of these symptoms are not the result of more spores coming in those last 10 days. They were here. Now it's not on the previous slide. If you go back in your memory, they were here when we had that one lesion. They just had not yet matured to the point of producing those symptoms on the leaves. So keep that in mind. As a plant pathologist, I go out and I see green fields, and our eyes deceive us. Everything looks lush and green, and these plants are already infected, but yet they've not yet started showing those symptoms. So keep that in the back of the mind as you're scouting these fields. So 21 days after that R3 application date, if that disease pressure and the environment is conducive for this continual development of the disease, we can just have this explosive growth of this pathogen. Now, again, this is kind of going back. Not every field will have the same level of environment. Now, this is kind of our honey hole from Frogeye leaf spot research that we do. This is down in Shawneetown, so that you can see the Mississippi River. You see the Ohio River in the far background. So that's Kentucky on the other side of that river. So it's a pretty. We're in the river bottoms. It's pretty far south. And this is what the picture looks like sometimes when we go out and rate. We typically don't like to go and rate soybean diseases, especially things like Frogeye leaf spot, before 10 o'clock in the morning. You get to the field, and that's not rain. That's the extended dew period. That's the fog still trying to burn off in the morning. So you do not have to have heavy rain or necessarily center pivot irrigation to drive this epidemic. Those areas that tend to be in the river bottoms, those areas that whenever we have these extended dew periods, we can see quite a bit of staying power with this disease and also infection. So this is the purpose of this talk is not to compare all potential chemistries for who's better at controlling this disease. Now, if you want to look at that data, you can go to the CPN website. They have a chart that goes through all the different the fungicides and talks about that. We do quite a bit of that screening. The purpose of this is just to show how explosive this disease is, even with some of the best chemistries. So these this particular comparison, we have the non-treated, we have Revitec, which would be 3 modes of action, and then we have Quadris Top. This was data that was collected in 2018. So at that time, that was one of the leaders in the market. That one has 2 modes of action as well. It's still a very good fungicide for controlling Frogeye leaf spot. And so this is rating the lower canopy. So this is another complexity with this pathogen is you don't just rate what you see with your eyes, and you kind of peel back. That tells us what was going on with the epidemic at the time of application. And you can see that both of those fungicides did a fantastic job. Now, our disease severity scale is 0 to 9. So 40 days after that R3 application, we're already at a 3. So it'd be 30% of our leaf area is covered with this pathogen in the lower canopy. In our upper canopy, this is where it gets kind of interesting. From 40 days after application, 47 and then 54, we see that even with the best chemistries that we have, this pathogen is starting to make a lot of inroads in that upper canopy. So the goal here, when you look at 54 days, that looks like a complete failure of fungicides, but that at that point, the yield has been made and has been protected. So, but that just shows you the explosiveness of this pathogen and why it's important to use multiple modes of action. If I had a single strobilurin, it would be matched up nicely with the non-treated in that environment. So it's very important to have multiple modes of action. I'm running out of time, but I know if I don't show the soybean yield thing, people will up. So at the R3 app, we see that both of those fungicides significantly protected yield compared to that non-treated. I didn't show the foliar data for the R5 app. Revitec worked similarly in both of those application timing. Interestingly, the Quadris Top did a little bit better with that later application timing. So we see that sometimes with different fungicides in different environments where they may perform a little bit differently depending on the application time. The last one, this one would be the one that would be, hopefully we never get to this situation in Illinois. This is a real thing in the South where they might look at both applications at R3 and at R5. And we look and we see a increase there at our Revitec at that R3 and R5 timing and with quadrus over the application of the R3. So there's a added advantage of that application at that time. Again, not across all fields in Illinois, but what we would hope that we never get to that situation where in all of Illinois, we're dealing with an aggressive pathogen in a way that we would have to have two applications of fungicide to protect yield. How do we not get there? It's using more than just fungicides to control this disease. It's starting with what's the field history, what did we plant there last year, what's the rotational history, and then also our seed decision is very, very important. Let's see, I'm going to skip through Septoria and get to our stem disease. Now, one of the things that if you've ever done fungicide work on your farm, or if you're a consultant or you work in industry, we've all seen where sometimes the non-treated soybean plots sometimes have a different color. Sometimes they're a little grayer. And when we're treated with these fungicides, especially where they have strobilurians in the mix, we will see sometimes this brown or healthy color compared to the non-treated. And if you look, I don't know about the lighting here on this slide, it's perfect, but I don't know about on screens, but from where you're sitting, but you can see one of the location there at Shawneetown where we're harvesting. You can pick out those non-treated blocks in that field. And so you can see that from the combine. It's sometimes not as easy to see when you walk in. So if I go in and take a picture, this is showing the non-treated, a lot of growth on our stems and our pods. And then this would be headlined R3. So this was a strobilurian alone at this location for this, at least one of these treatments. So a pretty big difference just with a single mode of action at that R3 timing. It can have a big influence on what stem pathogens are there, and also their incidence and also their severity. And some of the most common ones that we see is anthracnose. Now, typically for anthracnose, we see them show up late. So that's most of what we see, at least in Southern Illinois. We also see it up in Central Illinois, this discoloration both on the pods, but also the stems at maturity. If you ever get into a situation where you're actually seeing it showing up on green tissue on your pods and also in your leaves, that is an intense disease environment that we typically do not see that in Illinois, and hopefully we don't see that in the future. But this pathogen shows up way early in those reproductive stages, and it has a very, very long latent period. Typically, we don't know it's there until the plants are mature. And as I said, it's usually widespread. So those stem diseases that are most common that we see is anthracnose. Also, sometimes we can confuse it with pod and stem blight. It's very common too. So usually there's going to be a split. It's either going to be either or. In the state, sometimes you might see both of them on the same plant. And then charcoal rot. Last year was an explosive year for charcoal rot. So that's a soil-borne organism that infects our roots soon after planting. But as we get into real hot and dry conditions, it just continues to grow into the vascular tissues, and it exacerbates the drought that your plants are suffering from, or the dry conditions. No resistance is known in any crop to this disease. Fungicide seed treatments really don't have that much efficacy against this pathogen. In-furrow fungicides have really failed to take hold on this. This is one of those diseases that you don't really talk about at field days too much, because the only option to really lessen the severity of the disease in the season is to irrigate. So that's just not really that possible, you know. So it's a very challenging pathogen. This pathogen is focused by universities and companies. It's not that people haven't been looking at it, but across all crops, this pathogen is a challenge. It infects corn, soybean, wheat, whatever we grow in our fields is a host to this pathogen that causes charcoal rot. We do not have resistant varieties available to those top three, but we do have resistant varieties to some other major stem disease pathogens and diseases: brown stem rot, stem canker, and also Phytophthora canker. So that is, most of that would be managed not in the season, but in our variety selection. And most companies got a good taste of a flare up of stem canker. My years are all mixed up with COVID. So my reflection on that happened 2, 3, or 4 years ago, but I'm just gonna say within the last 4 years, we've seen a rapid uptick in stem canker from the Southern Illinois, the tip of Southern Illinois, all the way up into the far reaches of Northern Illinois. Usually what happens is those things come alongside with new germplasm that's coming out, but also maybe some changes with the pathogen and also the environment. They come together at the right point in time to where it's from one end of the state to the other, we're all experiencing. So that's a pathogen that requires a lot of respect, but it's also primarily is going to be managed with resistant varieties. Even though it's a pathogen that's producing these spores that are attacking our stems that then lead to cankers, it's the timing of when you get the fungicide out to protect those stems at the time that those spores are moving is not well defined. So somebody might have great success and you could repeat everything they did, but by spraying by a day or two different or have a different planting date, and you may not have the same level of success as your neighbor. That's how erratic this disease is in terms of trying to control it with fungicides. So very, very difficult one to make a fungicide recommendation on that one, even though fungicides are efficacious against the pathogen. It's just the timing is very difficult. So 5 minutes. So we're 11:50, so we got 4 minutes. So this last thing I wanted to touch on about the stem diseases, we have a new project with the Illinois Soybean Association, and this is a collaboration between plant pathologists at SIU and entomologists at the University of Illinois. And so we've some of the points of this project so far is this area of the stems and what people are experiencing. We needed help with this. And so ISA helped us develop a survey of farmers. And so farmers participated in that survey, provided a lot of information. I'm not skipping over that. I'm just going to point you to Danilo, one of our grad students, has a poster that's going to talk a lot about what not only the farmers said they were experiencing in their fields with regards to stem diseases, but also some of the early results and some of the samples that they also sent us to our labs. So that's some very interesting information, very excited about this project, some of the pathogens that we're able to not only capture out of those tissues, but also maybe some of the next steps in some of the research that would be needed for those. We also have our entomologist colleagues conducted a survey across the state for various insects, but also primarily to have some sites to determine, to make sure that we've still not yet have infestation with the gall midge, but also with deck T stem borer, which is a major issue from central Illinois to southern Illinois. Not every single year. Last year we had a rapid harvest, so sometimes we don't see some of the effects of that pathogen in those kind of environments. But if we have an extended harvest window, weather and things of that nature, sometimes that's when you start seeing the effects of this insect that attacks the stems. Many of our locations, last year we do surveys, if you took a look at 20 plants, you would find at least half of them would have that little deck T stem borer in those plants. So direct yield losses sometimes not so great if you can harvest, but if your plants start toppling over, I'm not sure if I turned something off with that or not. I do have some chats over here, so she's going to manage those. Okay, so I'm going to wrap up with that and see if we still have 11:52. We're doing great. So the timing, so pressure's off a little bit. I agree a little bit. I think go to 11:57 and stop talking. So I'm going to quit talking and answer any questions. Yes, sir.
Hold on 1 second. You got to get you been talking to the mic.
Jason, if a soybean variety offers stem canker resistance, can you talk a little bit about the difference between northern and southern stem canker? And will that cover both?
That's an excellent question. So the question was, if we have a variety that has a stem canker resistance, would it have protection against both northern and southern stem canker? Usually no. Now, I'm not familiar with some of the, maybe the most recent germplasm improvements in that area that maybe some of the companies might have access to. But typically the resistance, it would be either to southern or northern. There is quite a bit of resistance in the germplasm for the southern stem canker. That's a terrible name because we have that pathogen in Illinois, and it's not due to Southern Illinois. It's typically, it was more of a southern pest, but it is widespread in Illinois. Northern stem canker is a different, is caused by a different pathogen, close related, but there's not as much resistance to northern as there would be southern. But they will, if you have a mixture of those in an area, the resistance is not going to provide protection against both.
I have a question from an online attendee. That they've seen a lot of the varieties in soybeans in northern Illinois that they never usually see. Are any fungicides good to fight this pathogen?
Many of the strobilurins mixed with another AI would have an impact on this particular pathogen. I made a mistake. I was supposed to repeat the question for the online audience. So anthracnose, more presence in northern Illinois in recent times. Does foliar fungicide have activity against that? And they would, but you would be spraying in the absence of seeing disease. And so that, that does, if everybody does that, we run into problems down the road. You see, we have strobilurin resistant isolates of Frogeye leaf spot because we fed that pathogen one chemistry over and over in Western Tennessee primarily, and in many other parts of this country. And we now have a pathogen we can no longer manage with that mode of action. So typically you could get response to that, but that's a great question. I'm going to probably sit here and just keep mumbling words, but it's they would have activity, but advice of spraying every single field in Illinois just for the potential of the pathogen, it has a lot of risk associated with that as well.
Red or root rot or crown rot, this started there. A lot of conversations I had with growers. Research I've done is continuous forward. Are you at Southern Illinois, are you aware of any trials that are going on right now? We're looking at possible seed treatment controls or something in the road?
That's a great question. So red crown rot has recently been identified in the state of Illinois and also in other locations here in the Midwest. I will say when I started in Illinois in 2019, the other plant pathologist at SIU at the time, John Russell, he offered students $1,000 if they ever could find that pathogen. When he worked on it in Louisiana, he said it would never ever move out of the counties that produce, or not in Louisiana's parishes, but counties that produce soybeans that are not touching the Gulf of Mexico. That was the environment that was needed for that pathogen 2019 early. So something has happened to not only allow that pathogen to move, but also operate and infect and be sustained over winter in our environments. Nathan Klecheski, used to be at the University of Illinois. I know has done a lot of work on this pathogen. Carl Bradley at the University of Kentucky is working on it. It's now been found in counties in Western Kentucky. There are some work with some active ingredients that do show a little bit of activity. Does it control it? Maybe it's looking at it as a seed treatment option, but I can't speak to which products and which products. I mean, I know what they found, but I don't think it's been published, and I don't want to get too far ahead of what they've seen. But that's encouraging that there's some fungicides that seem to have some activity against. To my knowledge, there's no known resistance for that pathogen.
I think we've got time for maybe one more question. It is going to push a little over, but if we've got one more question, the audience can make that happen.
Before she turns it off, please, Leo's passing out these books. Any other books left on that table, please take those individually. They're not that heavy, but to pack them home, that's a lot. And when I get home, I have to think about the amount of ice that we had. It's crazy. The weather's worse down there than it is up here, but that was not easy to load those in that suitcase in the back of my truck. So make sure you take these resources, and first come, first served on these up here too, even the disease compendium. So thank you.